Hello students, this is Vinaya ma'am here with colligative properties. To understand colligative properties, we are considering a solution that is salt in water. What type of substance is salt? Sodium chloride is a non-volatile solute. When it is added to water, that is a volatile liquid, you find that many changes happen. What are the changes that are happening? The physical properties are changing considerably, and those physical properties are dependent on the amount of salt that is dissolved and not on its nature. Such type of properties are coming under what is called colligative properties. What do you understand by colligative properties? These are all the physical properties which depend on the number of solute particles and not on its nature. For example, what all changes happen when salt is dissolved in water? You find that there is a relative lowering of vapor pressure of the solvent, which will result in elevation in boiling point, depression in freezing point, and osmotic pressure. We'll see it one by one. What is meant by relative lowering of vapor pressure? I told you, whenever we are dissolving salt in water, the change happening here is the vapor pressure is decreasing. Here, you can consider the solution made from water. First, we consider water in the first beaker. You find that the molecules that are present on the surface is capable of changing into vapor. So that contributes to the total vapor pressure. So you find that vapor pressure of pure solvent is always greater, and that is denoted by P0A. Now, when salt is added to water, you find that salt particles also occupy the surface. With a result, the amount of vapors that are produced will be lesser on above the solution. As a result, you find that the vapor pressure will be considerably reduced. So, you find that P0A is always greater than PA. For solutions, you find that Raoult's law is applicable. That is, PA is proportional to the amount of the solvent. So, PA is proportional to XA. That is, PA is equal to P0A into XA. What is the change in vapor pressure? That can be calculated from P0A minus PA. On substituting, we get the value as P0A into 1 minus XA. For binary solutions, you have learned that XA plus XB is equal to 1. So, you find that change in vapor pressure will be changed to P0A into XB. So, the relative lowering of vapor pressure P0A minus PA divided by P0A becomes equal to XB. What do you understand by XB? It is the mole fraction of the solute. Fraction of the solute can be calculated using the formula NB divided by Na plus Nb. Here we are taking a dilute solution. So the amount of solute dissolved is comparatively less. That means Na value is much greater than Nb. So Xb takes the form Nb divided by Na. Reason? Nb is negligibly small compared to Na. On substituting the values of the number of moles, you get the formula as Xb is equal to WbMa divided by WaMb. Now, what change happens as a result? You find that when vapors are decreasing, vapor pressure decreases. With the result, the boiling point is also affected. What do you understand by boiling point of a liquid? It is the temperature at which the vapor pressure of the liquid becomes equal to atmospheric pressure. At this temperature, the vapors produced will be maximum. When a non-volatile solute is added to a volatile solvent, the amount of vapors are decreased considerably. We find that amount of volatile molecules present at the surface of the solution will be less than that of the pure solvent. With the result, you find that it takes longer period for the solution to reach the atmospheric pressure, and so there is an elevation in the boiling point of the solvent. The elevation occurs here. We can just represent that in the form of a graph. On observing the graph, you find that the first curve that is drawn is that of the pure solvent. When it reaches 1 atmosphere pressure, the corresponding temperature is T0B. For solutions, we have told that the vapor pressure is considerably less. So, on heating, you find that it reaches 1 atmosphere mark only later, and the value given is TB. So, what is the change in temperature? That is TB minus T0B, and that is found to be proportional to the concentration of the solution. It is dependent on the amount of solute that is dissolved. That is, delta TB is equal to Kb into m, where Kb is known as the ebullioscopic constant, otherwise known as molar elevation constant. On substituting the value of molality, we get the equation as delta TB is equal to Kb into 1000 Wb divided by Mb into Wa. Now we shall see what happens in the freezing point. What do you understand by freezing point? Freezing point of a liquid is the temperature at which the liquid starts solidification. It is otherwise known as the fusion temperature. It is the temperature at which the vapor pressure of the liquid becomes equal to the vapor pressure of the solvent. At the particular temperature, you find that both the liquid and the solid coexist. This we will just depict in the form of a graph. Here, on checking the graph, you find that the pure solvent, when it reaches T0F, there is a sudden break in the curve. There occurs a phase change, that is, it changes from liquid state to the solid state. So the moment it changes from the liquid phase to the solid phase, that particular temperature is known as the freezing point, and that is denoted by T0F. Now take the case with solution. We know that solutions are having vapor pressure less than the corresponding solvent. With the result, it takes longer time to freeze. That means it requires a more cooler temperature so that it gets solidified. So that value is TF. So what is the change in temperature? That is, T0F minus TF becomes equal to change in temperature. Delta TF is equal to T0F minus TF, and that is proportional to the concentration of the solution, which results in the equation delta TF is equal to Kf into m, where Kf is known as the molar depression constant or cryoscopic constant. With the result, we get the equation delta TF is equal to Kf into 1000 Wb divided by Mb into Wa. Here, Wb denotes the mass of the solute, Mb is the molar mass of the solute, and Wa is the mass of the pure solvent. Here, you find that the depression in freezing point is dependent on the amount of the solute that is dissolved and not on its nature. So, this is an example for a colligative property. When a non-volatile solute is added to a volatile solvent, we have learned that the vapor pressure is decreasing. So solidification happens at a lower temperature than that of the pure solvent. With the result, there occurs a depression in freezing point of the liquid. Now coming to the next process, which is known as osmotic pressure. To understand what is osmotic pressure, first we should know what is a process called osmosis. The process of osmosis is the movement of solvent molecules from a region of high concentration to a region of lower concentration through a semi-permeable membrane. What is peculiar about semi-permeable membrane? It helps only certain type of molecules to pass through it. Experimental setup shows that we have taken pure solvent water in a beaker and we have inserted a tissel funnel filled with salt solution, which is separated by a semi-permeable membrane. The semi-permeable membrane can be either cellophane or cellulose acetate or egg membrane. You find that it allows only certain molecules to pass through. So, when we are inserting the tissel funnel containing the salt solution inside the solvent molecules, you find that the amount of solvent molecules are more outside the tissel funnel. So, the movement of solvent molecules happens in the inward direction, that is from a region of higher concentration through the semi-permeable membrane into the region of lower concentration. With the result, there is a capillary rise in the salt solution. That occurs due to osmosis. In order to prevent this process of osmosis, we apply a hydrostatic pressure on the solution side, and that pressure exerted is known as osmotic pressure. That is usually denoted by the letter pi. This pi doesn't have any connection with 22/7. This is just the way we depict osmotic pressure, and that is proportional to the concentration of the solution and the absolute temperature, with which we can just equate the equation as pi is equal to R into Ct, where R denotes the universal gas constant, C is the molarity of the solution, and T is the absolute temperature. On substituting the value of molarity, we get the equation pi V is equal to NBRT, where pi denotes the osmotic pressure, V is the volume of the solution, Nb is the number of moles of the solute, R is the universal gas constant, and T is the temperature. So pi V is equal to NBRT is the Van Hoff's equation for representing osmotic pressure. So this equation and its work related to osmosis fetched the first Nobel Prize in chemistry for Van Hoff. You find this osmotic pressure and the process of osmosis occurs in many situations. You find that whenever we are putting raw mango in pickles, or if you are salting the mangoes, what happens there? The size of the raw mango will decrease or it will shrink. Why does it shrink? Because you find that the water content present inside the raw mango is more compared to the salt solution outside. With the result, exosmosis results, and you find that the size of the raw mango will shrink. What do you understand by osmotic pressure? It is the hydrostatic pressure exerted on the solution side just to stop the process of osmosis. What are the applications of osmosis? As told earlier, raw mango placed in brine solution will shrivel. Withered flowers, when placed in fresh water, becomes revived. You might have heard of people who are having high blood pressure. If they take too much of salty food, that result in puffiness in the body. Why this puffiness occurs? Because there occurs retention of water inside the body, and this medical condition is known as edema. More applications of osmosis. You find that in olden days itself, there is a way of preserving meat and fish by salting it. What happens during salting? You find that all the moisture content is removed, and that occurs due to osmosis. At extreme concentrated conditions, you find that is the drastic conditions, the microbes and fungus cannot survive. The same thing happens when we are preparing jellies and jams. We are adding too much of sugar in that. So there too, in concentrated solutions, the pathogens cannot survive. Another important property that is used is desalination of water, where we use the process of reverse osmosis that we will discuss in detail. And in diluting injections with 0.91% brine solution, this also we will discuss in detail. Take the case with desalination of water. What do you understand by desalination? Removing salt from seawater. So, in desalination, we are taking the seawater and is separated from fresh water using a semi-permeable membrane. So, the experimental setup where you are doing desalination consists of a compartment which is divided by a semi-permeable membrane. Usually the semi-permeable membrane is coated with cellulose acetate. When we are applying an external pressure greater than the osmotic pressure, the solvent molecules from lower concentration region is forced to move to the high concentration region. So this is the reverse of osmosis, and that process we call it as reverse osmosis. This is the way in which the Gulf countries meet the want of drinking water. Now, before moving on to the next point, first we will see that different types of solutions based on osmotic pressure. We have three different types of solutions. One is called isotonic solution, the other one hypertonic, and the third one hypotonic solutions. When do you say that the solution is isotonic? Two solutions are said to be isotonic when both of them have equal osmotic pressure. In such a condition, when in the two solutions we place a cell, there is no change in the size of the cell. Whereas if you are placing a cell inside a hypertonic solution, the water from inside the cell will be moving out, which resulting the shrinkage of the cell. So what do you understand by hypertonic solutions? These are solutions which have high osmotic pressure. Coming to the next type of solution, where you find that the osmotic pressure is very less. In such a case, when we are placing a cell inside a hypotonic solution, the water from outside enters into the cell, with the result the cell gets bulged and finally it collapses. Why we have discussed this is to understand what happens when we are taking intravenous injections? So here we find that the blood cell is having a concentration which is equal to 0.91 sodium chloride solution. So when we are placing a blood cell inside 0.91% sodium chloride solution, the cell will remain in the same size. That is, no water is going out from the cell or getting into the cell. But when we are placing the blood cell inside a hypertonic solution, that is having higher concentration than 0.91% sodium chloride, so that is exerting high osmotic pressure. So with the result, you find that water content from inside the cell moves out, with the result that cell will shrink in size. The opposite happens in the case of hypotonic solution. Hypotonic solution will have less amount of salt present, and with the result you find that water from the higher concentration region move into the cell, and as a result the cell gets bulged and it collapses. This happens when we are taking intravenous injections. In the case of intravenous injections, if you directly inject the medicine into the human body, it may either result in shrinkage of the cell or bursting of the cell. In order to avoid that, we are maintaining it in isotonic condition. How is it maintained? We are diluting the injections with 0.91% sodium chloride and then administering it on the patient. With the result, there is no shrinkage or bulging of the cell.